Hey there, science fans! Welcome back to another exciting journey into the world of science. Today, we're diving into the fascinating world of gel electrophoresis, a technique that might sound complex but is incredibly interesting once you get to know it. This powerful technique allows scientists to separate molecules based on their size and charge, making it an essential tool in many scientific fields. Imagine a microscopic racetrack where molecules zoom along, competing to reach the finish line. Each molecule moves at its own pace depending on its size and charge. That's what gel electrophoresis is all about. It's like a high-speed chase but on a tiny scale. This technique is like magic, but it's not magic, it's science. And it's a science that has revolutionized the way we understand molecular biology. It's used in countless fields, from medicine and genetics to forensics and evolutionary biology. Each field relies on gel electrophoresis to uncover vital information. Ready to learn how it works? Let's go. We'll break it down step by step so you can see just how amazing this process is. Gel electrophoresis is like sorting a bowl of candies by their size, except we're sorting molecules and they're much smaller. Imagine trying to sort grains of sand by size. It's a fundamental tool in molecular biology, helping us understand the building blocks of life. Without it, many of the advances in genetics and medicine wouldn't be possible. So next time you hear about a breakthrough in genetics or a new forensic discovery, remember that gel electrophoresis played a crucial role in making it happen. Stay curious, keep exploring, and never stop asking questions. Science is all about discovery, and there's always more to learn. Before we start our molecular race, we need to set up the racetrack. This racetrack is crucial for the journey our molecules will undertake, and it all begins with careful preparation. This is where the gel part of gel electrophoresis comes in. The gel serves as the medium through which our molecules will travel, and its preparation is a meticulous process. The gel is like a jello-like slab, often made from agarose, a seaweed extract. Agarose is chosen for its unique properties that make it ideal for creating a stable yet flexible matrix. We pour a hot liquid solution of agarose into a mold, and as it cools, it solidifies into a porous gel. This transformation from liquid to solid is essential for forming the structure that will guide our molecules. These pores are like tiny tunnels that our molecules will navigate. The intricate network of pores is what allows the gel to act as a sieve, sorting molecules based on size. The size of the pores can be adjusted depending on the size of the molecules we want to separate. This customization is key to achieving precise separation. Smaller pores for smaller molecules, and larger pores for bigger ones. This flexibility allows scientists to tailor the gel to their specific needs, ensuring optimal results. It's like choosing the right size sieve to separate sand from gravel. Just as a sieve sorts particles by size, the gel sorts molecules, making it an invaluable tool in molecular biology. The gel acts as our molecular sieve, allowing us to separate molecules with incredible precision. This precision is what makes gel electrophoresis such a powerful technique in scientific research, enabling us to analyze and understand complex molecular structures. With our gel ready, it's time to introduce our contestants, the molecules we want to separate. These could be DNA fragments, RNA strands, or even proteins. We mix our samples with a special dye that makes them visible and helps us track their movement. Using a micropipette, a tool that allows for precise measurement of tiny volumes, we carefully load our samples into small wells at one end of the gel. These wells act as starting lines for our molecular race. Think of it like lining up runners at the start of a race. Each well contains a different sample, and we're eager to see how fast each molecule can go. Now comes the exciting part. We submerge the gel in a chamber filled with a special buffer solution that conducts electricity. This chamber is connected to a power supply, and when we flip the switch, we create an electric field across the gel. Remember that molecules can have an electric charge? DNA and RNA, for instance, have a negative charge. So, when we apply the electric current, these negatively charged molecules are attracted to the positive electrode at the opposite end of the gel. It's like a magnet attracting metal objects. The positive electrode acts as a molecular magnet, pulling the negatively charged molecules through the gel, and the race is on. Section 5. Size matters. How molecules separate. As our molecules race toward the positive electrode, they encounter resistance from the gel matrix. Smaller molecules can navigate the tiny pores more easily, while larger molecules get slowed down, like trying to squeeze through a crowded room. This size-dependent resistance is the key to gel electrophoresis. Over time, smaller molecules will travel farther through the gel, while larger molecules will lag behind, creating distinct bands of separated molecules. Imagine a group of people of different sizes running through a field of tall grass. The shorter folks can weave through the grass more easily, while the taller folks get tangled and slowed down. Section 6. Seeing is believing, visualizing the results. After a while, we turn off the electric current and carefully remove the gel from the chamber. But how do we see the separated molecules? Remember that dye we added to our samples? This dye fluoresces under UV light, meaning it glows when exposed to ultraviolet radiation. We place the gel on a UV transilluminator, and voila! The separated molecules appear as distinct bands, each band representing molecules of a specific size. For proteins, we often use different staining techniques such as Q-Massey blue staining to visualize the separated bands. It's like revealing a secret code, with each band telling us something important about the molecules in our sample. Section 7. DNA Detectives Gel Electrophoresis in Action. Gel electrophoresis is an indispensable tool in DNA analysis. One of its most famous applications is in DNA fingerprinting, used in forensic science to identify individuals based on their unique DNA profiles. By comparing the banding patterns of DNA samples collected from a crime scene to those of suspects, forensic scientists can determine if there's a match, helping to solve crimes. It's like comparing fingerprints, but at the molecular level. Gel electrophoresis allows us to visualize the subtle differences in DNA sequences that make each individual unique. Section 8. RNA under the microscope. Unraveling the Mysteries. Gel electrophoresis isn't just for DNA, it's also a powerful tool for studying RNA, another vital molecule in our cells. RNA plays a crucial role in protein synthesis, carrying genetic information from DNA to the ribosomes where proteins are made. By analyzing RNA using gel electrophoresis, scientists can study gene expression patterns, identifying which genes are active or inactive in different cell types or under different conditions. It's like listening in on the conversations happening inside our cells. Gel electrophoresis allows us to eavesdrop on the molecular chatter of RNA, revealing the secrets of gene regulation. Section 9. Proteomics Powerhouse Analyzing Proteins. Proteins are the workhorses of our cells, carrying out a vast array of functions from catalyzing biochemical reactions to transporting molecules. Gel electrophoresis, often in combination with other techniques like sodium dodecyl sulfate, polyacrylamide gel electrophoresis, SDS page, allows us to analyze the composition and abundance of proteins in a sample. This is particularly useful in studying diseases like cancer, where protein expression patterns can be altered. It's like taking inventory of the tools in our cellular workshop. Gel electrophoresis helps us understand how proteins contribute to both health and disease. Section 10. The versatility of gel electrophoresis, a scientific Swiss Army knife. Gel electrophoresis is a true scientific workhorse, used in countless applications across a wide range of fields. From diagnosing genetic disorders to developing new drugs, its versatility is unmatched. Its simplicity, cost-effectiveness, and ability to provide clear, visual results make it an invaluable tool for researchers and clinicians alike. It's like a Swiss Army knife for molecular biologists, offering a suite of tools for separating, analyzing, and characterizing the molecules of life. Section 11. Conclusion. Gel electrophoresis, a cornerstone of science. Gel electrophoresis has revolutionized the way we study and understand the microscopic world. This technique, which involves the separation of DNA, RNA, or proteins based on their size and charge, has become an indispensable tool in molecular biology and biochemistry. Its ability to separate molecules with incredible precision has opened up countless avenues of research, leading to groundbreaking discoveries in medicine, genetics, and beyond. From diagnosing genetic disorders to identifying pathogens, gel electrophoresis plays a crucial role in modern science. As technology continues to advance, we can expect even more innovative applications of this powerful technique, further illuminating the secrets of life's molecular machinery. The development of new electrophoresis methods and equipment continues to push the boundaries of what we can achieve in the lab. So the next time you see a glowing gel image, remember the incredible journey those molecules have taken, racing through a microscopic obstacle course to reveal the secrets they hold. Each band on the gel represents a step forward in our understanding of biology and chemistry. Gel electrophoresis is more than just a technique. It's a window into the fascinating world of molecules. It allows scientists to peer into the very building blocks of life, providing insights that were once thought impossible. This cornerstone of science continues to be a beacon of discovery and innovation.